To the fourth ReMO seminar. Tonight we're going to hear about the role of particles in the biological carbon pump by Morten Iversen. And I think everyone knows Morten, and I don't have to add much, Morten. The screen is yours.
Thanks. That's a compliment. Yeah, so I'm very happy that you are willing to listen to a talk that I put together about the role of particles in the biological carbon pump, which was a talk I also gave at the Gordon Research Conference 2 months ago, I think. And there are parts about respiration, but I'm quite sure that there's probably also other parts in here that might be of interest to some of you, and if not, then I hope you will still enjoy it. And I will still give the very brief introduction to the carbon pump, even though I think everybody knows it, but I always look at the carbon pump. I think there's two layers. We have the photic zone in the surface ocean and the aphotic zone below that, typically below 100 meters. As you all know, CO2 is fixed into organic carbon by phytoplankton photic zone, which can then, of course, be grazed by animals or respired by bacteria in the surface ocean. That will immediately release the CO2 and that will go into the atmosphere. But when we talk about the carbon pump, we are more interested in these particles or the aggregates that are formed. And we really put it down to the most simple forms of aggregates. We have two types. One is clusters or aggregates of phytoplankton, which we tend to call marine snow, even though marine snow is defined by being bigger than 0.5 millimeter. And sometimes leads us to figure out what we call all the aggregates that are smaller. Normally we just call them phytoplankton aggregates, but it's basically one whole range of particles that can be anything from just a few phytoplankton up to meter-large aggregates. For instance, in the Adriatic Sea, we have these meter-big aggregates that are sitting and sinking very slowly. And then another very important aggregate is the zooplankton fecal pellets. And that's basically the two types of aggregate I focus most on, the fecal pellets and the marine snow. Of course, when we talk about export flux, we want those to sink out and actually get into the deep ocean or into the aphotic zone, which we have below typically 100 meters. And what we tend to do is we measure the PUC flux in the depths below the photic zone, so anywhere from 100 meters and down to many thousand meters. And here I have plotted it over time. This is actually a real time series from Cape Blanc, 30 years, where you can see we have some peaks and we have some valleys. And what we're trying to figure out is what is actually causing these peaks in carbon flux and what is causing the periods where you have low carbon flux. And really, if you put it to the very simplest form, you can say that export flux is determined by degradation of organic matter, so turning organic matter back into CO2, and by the sinking speed of these particles. And sinking speeds, when you look at a vertical water column, is basically determining time you have to respire organic matter back into CO2. And here we have a little video that we made with an ROV and remotely operated vehicles, so an underwater camera, where you can see these particles are sinking slowly down through the water column. And for now, I'm just using it as a nice background for the outline. So tonight I will tell you a little bit about the biological carbon pump, and then we will go into the different attenuation processes through the water column. I've divided that into two main processes, one being what is the attenuation of this carbon flux via zooplankton feeding on aggregates, and also what kind of processes are involved in the feeding, such as disaggregation, respiration, and repackaging of the organic matter. And then we will go a bit more into the microbial colonization and degradation of aggregates. And here I'm thinking that Gerhard will lead to some nice discussions about what is actually current state of knowledge or accepted and what he has found recently. And then finally, we'll look a bit more into nutrient recycling and stoichiometry of these aggregates and try to see if we should maybe start considering attenuation processes not just as a decrease of carbon flux, but it could also increase the efficiency of the export of organic matter via recycling nutrients and changing the stoichiometry of the aggregates, which is in some models, but most models don't actually consider change of stoichiometry of aggregates. And then if we talk about the carbon pump, I think 5 years ago we could just talk about settling of aggregates more or less. But recently everybody has become much more aware that it's not just sinking, gravitational settling of aggregates that is driving the carbon export. But we have ocean mixing, which is not just important for CO2 being mixed down, but also for how particulate organic matter is sinking down. And then also the migrant pumps has received a lot more attention recently, where we have seasonal migration in the Arctic Ocean, where copepods with a lot of lipids swim down and respire lipids during the polar night or during winter. And also the daily migration of zooplankton, which Javier is working a lot with, and Santiago also. And of course, when we really talk about sequestration of organic matter, it depends how deep organic matter is sinking down, and we can only really talk about things that reach deeper than 1,000 meters to really have significance in how long carbon is stored away from the atmosphere again. So Philip Boyd made a nice overview where he compiled some of the newer research. I don't actually know that I have a green line, so I'm not really sure you can see the references here. But otherwise, I can always send you all the slides after. He divided it into the gravitational carbon pump. This is what we have typically been considering to be the biological carbon pump in the past. Then he added some other pumps, which he termed injection pumps. And here he had eddy subduction. So when you really have water that is transported down physically, so subduction of water, which can bring single cells, but also technically increase the terminal settling velocity, because you have a particle that sinks, but then the water also sinks. So we can have a very efficient way of bringing carbon down to depths. These typically work in the upper ocean, so 250 or 400 meters. We have the mixed layer pump, which is when you have the winter mixing, you can have single cells coming down. And I think Giorgio can probably say a lot more about that also, where single cells can really be brought down during winter. And if the mixed layer in winter is increased and they get trapped down there, and then you have the spring mixed layer coming up, then you can also have a way to store organic matter in the deep ocean. At the moment, we don't really know how fast it happens, and we are also quite unsure about where this goes down to. And then we have the mesopelagic migrant pumps, which can be the daily migration of zooplankton that swims up during night, feeds in the surface, swims down to depths below the mixed layer, and then produce their fecal pellets. And that can be a very active way where they can basically short circuit the whole microzooplankton and zooplankton that can feed on these particles, and they can get fecal pellets with fairly high sinking velocities down to 200 or 1,000 meters. It depends on the organism. And they can produce the pellets, which will then sink down without being degraded a lot. Then, of course, seasonal lipid pump, which is in the Arctic, where zooplankton spends most of their time in the surface during the polar day, producing these lipids, and then they spend the whole polar night at depths respiring the lipids and living from that. And if you look at the different pumps, we can see here we have the mixed layer pump, MLP, which is the orange here, which tend to happen around spring. Then we have the large-scale physical pumps, which are basically also the conveyor belts, the large ocean circulation. and also the mixed layer pump in the winter, so that tends to be very efficient during winter, but it occurs during the whole, as all other seasons, and then picks up again end of autumn. And then you have the eddy subduction pump, which is mainly winter to spring and autumn to winter, and the seasonal lipid pump, which is during the polar night, so basically during winter. And then we have the mesopelagic migrant pump, which tends to happen after, when primary production kicks off and when zooplankton had a chance to really become effective in their amount and their grazing, but also in their migration. And this is typically in the temperate regions that is really efficient. That's why it happens somewhere in late spring and ends during autumn. And then of course you have the rotational pump. This of course happens to a large extent right before zooplankton kicks in spring, temperate regions, and then it becomes less important during winter. If you're actually comparing them, so they were doing a modeling study in Boyd et al. 2019, and they find if you combine all the particle injection pumps, then potentially you can provide an additional carbon flux, which is similar to what we have measured from thorium or sediment traps with the biological gravitational pump. One thing they didn't discuss so much in the paper was whether we are really measuring a lot of the injection pumps already using sediment traps or thorium, for instance, which I would argue that we could be doing with the mesopelagic migration pump. But that's something there hasn't been a lot of studies in this direction yet. We did, because we found the whole injection pump where you have basically basically the subduction, but also if we go back, so it's the large scale physical pump and the eddy subduction pump. So we were trying to look off Cape Blanc. This is an upwelling region where we have a lot of deep water coming up to the surface ocean, and then we have Ekman offshore transport. And here we wanted to see how much of we have 30 years of deep ocean sediment traps sitting offshore, one we call CB, euphotic, because it's more productive. And we have the CB mesotrophic, which is in a very low productive area. We wanted to see, could we actually explain a lot of the carbon flux that we are finding at these offshore deep ocean sediment traps via the production that happens on the very productive shelf, via this Ekman transport and slow sinking of particles. So we went to Northwest Africa here. This is Cape Blanc with Morocco and Mauritania. We were trying to use drifting traps and a lot of in situ optics, of course CTDs, and we sampled individual aggregates to measure the microbial degradation with marine snow catchers. And then on the shelf, we had a lot of lander systems, and we measured primary production and benthic primary production. I was trying to follow the resuspension to see how much of the carbon that is produced on the shelf could be transported offshore via Ekman and then slowly sinking down and actually landing in the deep ocean sediment traps, and whether our traditional view of just doing a vertical transport of settling particles is at all relevant in these upwelling regions. And when we did the camera profiles, we did find, so here we have, if you look at the scale, this is longitude, so it's compressed a lot, but we did find that a lot of materials seem to sink down following the slope. We also had some large particles that sank quite slow, was kind of transported from the shelf and offshore, and could be caught in our sediment traps. So we did see a very strong lateral advection off the shelf, and that was of material that was actually produced on the shelf, which ended up in the deeper ocean offshore. And we were trying to use the drifting sediment traps. So here you can see the measured in red, and then we were trying to calculate it using the microbial degradation that we found for depths below where the zooplankton were feeding a lot. What we found was the closer we were to the shelf, there was actually less calculated flux if we just assumed it was sinking down than what we actually measured. And the further we got offshore, the less of the predicted or calculated carbon flux was actually coming down. So basically it showed that if we look at the 400-meter traps, that we had a shallow export to 400 meters near the shelf. So these two stations were near the shelf. And that was explained, or that it was higher than what we could explain by things that was produced in the surface just above it. So there must be some lateral advection to have this higher measured flux than what we would actually expect would be coming down from the surface ocean. We then used a 30 years sediment trap time series. We found exactly the same here. So we had the flux at 1,000 meters. We used microbial degradation, which we had measured. We found that for all seasons, when we combined everything, we had higher flux at depths than what theoretically should be able to sink down. And this is something which we can discuss with Gerhard. Even if we doubled this, because obviously we took the aggregates up on deck, measured the respiration and saw how much should be gone, and measured the settling velocity. Even if we doubled flux that we found, that we still find higher fluxes. So it seems like something is definitely coming in from the side. So we have a very strong transport of shelf-produced organic matter, so primary production from the shelf, that is sliding down, following the water masses and landing in our offshore traps, which is something we haven't included in models so far at all. So we found that for the 3,500 meter traps at all seasons was a strong lateral input to the export flux. And when we did the whole budget for our cruise, so I'm not expecting you to really look into details, but we measured primary production at all depths, POC, standing stock, we used camera profiles and drifting traps and deep ocean traps. We could really see that there was definitely organic matter were produced on the shelf. Also because on the shelf we found it was quite shallow, only 60 meters. We had light coming down to at least 25 meters, but at times even deeper. So we had the particles sinking down, resuspension, microbial degradation, and the nutrients were released again. So we had quite a lot of both resuspension, but also nutrients that were released back up and which fertilized this primary production. And all that organic matter was transported offshore via Hickman. And it really led to that we had a lateral advection, shelf production, via these slow settling particles that contributed by 80% to the deep ocean carbon flux in all seasons between 1988 and 2021. It might be, if the microbial degradation is only 50% in the deep ocean, this is only 40%. It's still a significant part. And it's still really changing the whole. I'm probably preaching to the choir, but this way of looking at a vertical water column is something we have to be really, really careful about. I mean, we have to consider that stuff is coming from the side and being laterally advected. And when we measured the settling velocity of these particles, we found that we had a lot of really slow settling single cells or phytoplankton settling with less than 1 meter per day, but with quite, so 6.9 kilometers per day offshore advection. When we went between 100 and 500 meters, we had a huge amount of organic matter sinking with only 5 meters per day. And then we had this slope, following the slope down with around 35 meters per day of organic matter. And then, of course, on top of that, we have marine snow and the fecal pairs that are sinking more or less straight down. It seemed to be a substantial fraction of the organic matter that were really produced on the shelf and sinking offshore. And when we are looking at the efficiency of the carbon pump being completely offshore, we tend to look at this vertical view, which might be biased quite a lot by this lateral advection in eastern boundary upwelling areas. So we just went 2 months ago to test this off Ireland, which is not an upwelling region, to see if this is also a thing that happens in non-upwelling regions. But especially in the upwelling regions, we might have to reconsider the efficiency of the carbon pump based on the strong Ekman transport and self-produced organic matter. We also know that in the Arctic, we have, especially at the ice edge, when we have the meltwater coming, we have at Fram Strait, which is where we have our main research area between Svalbard and Greenland. This is basically where all the ice is exported out of the Arctic into the Atlantic, and it tends to melt in this region between Greenland and Svalbard. And as it's melting, we get the surface meltwater, and we have the polar water. Then we have the Atlantic water coming in. And right at the ice edge, we tend to get quite dense water that really sink down and can potentially transport a lot of organic matter down with it. So this is another of these mesoscale pumps where you have this strong subduction. And we went on a cruise 2 years ago. We did camera profiles. So this is a camera profile that we did where we were measuring total particle numbers. So we had the ice edge here on the left and the Atlantic water coming in on the right. And we saw right where the ice melt happens and we get this meltwater formation. We had basically particles that went down extremely fast. We saw this high number of particles down to 500 meters. And what we noticed was we had a very, very strong Phaeocystus bloom. And Phaeocystus, when it's not ballasted, doesn't really sink at all. What we found was that a large fraction of the total carbon flux we measured at 100, 200, and 400 meters with our sediment traps were actually made up by Phaeocystus, even down to 400 meters. So then we measured the settling velocity of Phaeocystus colonies in the laboratory, and we found that they were sinking with 0.5 meter per day, which would mean that it would take them 2.2 years to reach 400 meters. And these Phaeocystus colonies were fresh, happily photosynthetic, so it wasn't 2.2 years that they had been in the deep ocean. That was clear. And when we calculated the exact carbon flux, we had between 15 and 40% of the total carbon flux was contributed by Phaeocystus. So that really suggests that the subduction zones near the ice edge may be important sites for POC export at the moment. We all know that ice is disappearing, going away. So the question is, especially in the Arctic, we haven't tested this in Antarctic, but we went on a cruise called Cosmos last year in spring, and we did all the measurements, and we are calculating all the data at the moment to see, because we also found massive Phaeocystus blooms at the ice edge in the Antarctic. So this might not only be for Arctic, but for both Arctic and Antarctic. Basically, at the moment, we might have a fairly efficient carbon export in the Arctic because of these ice edge formation. What is happening when ice retreats and we only have ice in the central Arctic? Will there still be Phaeocystus bloom? Will it be more Atlantic? That's definitely something we need to look into in the future. But it was quite interesting to see that it would have taken 2 years, but we have healthy looking, happy Phaeocystus colonies at 400 meters, which shouldn't be down there. So it must be subduction that caused this highway of organic matter really just whizzing down within days. Just to say, it's not all these particle injection pumps. There was just a new study by Dave Siegel's group, Nowicki, I think it's pronounced, from this year actually. And they did a large modeling, a global modeling study, and they found that instead of having 50%, it was 30%. So it may be that the particle injection pumps are not as important as they were depicted in Boyd's paper from 19, but 30% is still a large contribution, especially if it's overlooked at the moment. So settling aggregates still seem to be important, whether whether it's 50% or 70%. And they were suggesting that 85% of all carbon flux was due to fecal pellets and only 15% due to marine snow, which seems a little incredible when you look at the numbers, because fecal pellets are important, especially in the Southern Ocean or in temperate regions. But if you go to low latitudes, it's primarily marine snow sinking down. So that's another thing where we really need to look into it. Should we really spend more efforts on zooplankton, or is it actually marine snow? And I think this is a seasonal thing when you're really studying it. I mean, you have different seasons right after the peak of the spring bloom in the temperate regions. You will have a lot more marine snow than fecal pellets. And I think it's quite interesting still to consider what is actually the processes we need to focus more on in the models. Is it fecal pellets or marine snow? And I think it's seasonal and it's regional, but it's something we really don't know. And just to put out numbers saying it's 85 versus 15 is of course dependent on what to put into the model, but I think these numbers are still up for debate, sure. We were then trying to figure out somehow if we could divide the water column into vertical processes, and for that we combined high resolution camera systems with sediment traps and have been doing this for the past 15 years. Of course, increased over time. When we got more money, we could build the camera systems as it is. Every time you get new equipment, your conclusions, of course, changes. And with the new camera system we have built, we can both see zooplankton and aggregates, but we also have the option that we get the camera, the pictures directly on board while the camera is in the water. And we can do stops and really look for specific processes. We can also change the zoom of the camera, so we haven't we've done it on the last cruise, but I didn't have the pictures ready. We can actually go down to 2 micrometer resolution, so we can now actually go from large zooplankton and down to single phytoplankton and down to 6,000 meters with this system, and then get the pictures on board while the camera is in the water. And our next plan is to actually deploy this camera system with mini marine snow catchers, so we can sample aggregates as we go down, and measurements on board. Of course, pressure is always an issue, so maybe Gerhard, we can discuss if there's any option to do some kind of pressure constant sampling. But basically what we are getting is we have the sediment traps, which measures actual flux, and then we can calculate the flux from the presumably settling particles. That's the next thing we are trying to figure out, if we can separate suspended to settling particles. But we can get this high resolution carbon flux, which when you look at it, you can kind of divide it into different processes where you have high attenuation in the surface, you have some kind of intermediate attenuation, and then in the deep ocean you have low but constant attenuation of the carbon flux. And that we can then calculate into carbon specific degradation, and we can start looking at really numbers of how much carbon from the settling particles are degraded per day. And we can do it for day and night, and then we typically find low carbon degradation during night in the surface, but high, during day in the surface, but high during night. We go below the mixed layer, find that there's no change between day and night, and in the deep ocean there's also no change, but it's much lower. And when we then look at the different processes that could be responsible for this degradation, if we start looking at this additional degradation that happens during night, then the most intuitive, which I think is also what people believe nowadays, is that it's due to zooplankton grazing that migrates up, which is again regional, because you do have regions in the Arctic, for instance, you don't have a day-night migration during the polar day. If you look at temperate areas, do tend to have that all the zooplankton comes up during night and feed. So we wanted to see, can we actually explain these, in this particular case, these extra 33% carbon degradation that happens during night by zooplankton. So we used the marine snow catcher to catch marine snow, of course, and we collected zooplankton, and then we used infrared light to film if the zooplankton would actually feed on the settling particles using two cameras, so we could get a 3D view of it. And here is a video from Fabienne Lombard, where we see a settling appendicularian house, and you can see all these Temora, it's a copepod, and they tend to swim horizontally through the water column until they can basically taste the tail of dissolved organic matter behind an aggregate, and then they follow this chemical trail until they're attached to the aggregate, feed a bit, and then they jump off. Because it's of course very dangerous to sit on a large particles which a fish could detect and feed on. And there are many copepods with many different strategies to detect, find, and feed on these particles, and that happens as they're sinking down, which causes disaggregation of the aggregates, and they're repackaging these aggregates into fecal pellets that would then sink faster, or they're just respiring it and degrading the particles or releasing nutrients even. This happens all the time as a particle sinks through the water column. And I think I have another example from Northwest Africa where we filmed Metridia feeding on a marine snow, and here you can see it kind of catches it, and then it seems to be feeding on the surface. There's also disaggregated, so you get all these small aggregates which sinks a lot slower than the mother aggregate. And this is basically what Giorgio has been looking at, and Nathan Briggs and all of those, to see if it's not just the feeding on aggregates that causes zooplankton to be important for attenuation, but it could also be the disaggregation, which leaves them to sink much slower and have much more time for microbial degradation. And here we have from the Arctic a Calanus, which is catching an aggregate, which is possibly more by luck that it found the aggregate, but you can definitely see it doesn't pass up the aggregate. So when it's swimming in, it's definitely catching it and it's feeding on it, and you can even see it's got slowly filling as it's feeding. So it's definitely a thing that they're feeding on these marine snow aggregates. And the question is, does this just attenuate carbon flux? Do they do vertical migration and produce compact packages via their fecal pellets which can sink much faster? Or do they release urea, which basically becomes ammonia, which can then be used by phytoplankton? So are they really fertilizing? Is this a good thing for the carbon pump or a bad thing? And this is what we are talking about next. And I think it's actually more efficient when zooplankton feed on aggregates. It makes a more efficient carbon pump than if we didn't have zooplankton. But we will get to that in a bit. So what we found was we used sediment traps between different depths in the water column, looking at the actual flux, and then we calculated the attenuation or the POC degradation from the sediment traps. We did the same with the cameras. And then we did the same for the zooplankton. So the colors are not great, but you can see the light one up here was one copepod called Pseudocalanus. The one that is a bit darker is one copepod group called Calanus. And then we have the microbial degradation. So this is looking at two copepods and microbial degradation in the Arctic. And they made up a significant part of the total flux attenuation just by considering large copepods and microbial degradation. This is without considering ciliates, dinoflagellates, and all the small copepods that exist. And just by having these, we could explain the majority of the carbon flux attenuation in the Arctic. This is completely different in the tropicals, where you have tons of different species. But we found that just by looking at Calanus and Pseudocalanus feeding alone, we could explain 60 to 67 of the POC flux, % of the POC flux attenuation. And if we included the microbial degradation, which we measured with microsensors on the aggregate, we could explain 77% of the total POC flux attenuation. So this is something you can actually fairly easily include in a model and start on, because there has been years of studies of these two copepods in the Arctic, and we really know their seasonal behavior. Another copepod, we then wanted to study the small copepods in the Arctic, Microcetella. This is a copepod that is less than 200 micrometers long, but extremely abundant. And here we were testing, how does it actually find food? So first we filmed them in an aquarium without any food, and we found that they were swimming up. And once they had been swimming for some time, they let themselves sink back down. But they didn't sink as long as they were swimming upwards, and they were always swimming upwards in spirals. And once they actually found a region with food, which we have here, they started swimming horizontally, because now they were swimming up. Oh, this is where all the particles are, and now we are swimming horizontally, and that really increased their chance of finding the food particles. And now we are in a buffet full of nice German sausages or whatever we have. And then they would swim horizontally and really detect the particles and feed on them. And whenever the particles had sunk out, they started doing the sinking again and the slowly upward swimming until the next vein of particle came, and then they would swim horizontally. So this is really how you can see that swimming behavior of these copepods really makes a difference to how they can find the particles. And what we saw was, here we used video plankton recorder, VPR, to film it. So the black here is marine snow particles, and the red are fecal pellets. And we could see that marine snow were formed, but they also didn't have a chance to sink out. So something was feeding on them. And when we then looked at these tiny copepods, but they're super abundant. So we often found 40 to 50 per liter, especially in the layer where we had all the marine snow particles. So it's really masses of these tiny copepods that are searching the water column, and once they find the depths where you have marine snow, they start swimming horizontally and really detecting the particles, breaking them apart, feeding on them, respiring them, etc. And we then were like, okay, but are they really feeding on them? So we did, I don't know if you can see, so the x-axis here is marine snow aggregate concentration. And because you can't really measure how much they feed because they're so small. So what we did instead was that we counted the fecal pellets when they were only offered marine snow. We definitely found a functional response where we could see the more aggregates there were, the more fecal pellets they produced. So this is a direct indication that they are actually feeding on these marine snow aggregates. And we found we could determine ingestion rates on marine snow of 0.26 microgram POC per individual per day, which is next to nothing. But considering the abundance of these, it actually made up a huge amount of the degradation of the aggregates. And then we did the same with krill fecal pellets. It was a bit lower, but again, they were actually effectively feeding on these particles. And basically, due to this high abundance, we could explain 20 to 100% of the flux attenuation just by Microcetella. Put that together with the 66% of the Calanus and Pseudocalanus, it really seems like copepods are quite important for flux attenuation in the Arctic. These are mainly coastal, so they don't really occur in the open ocean. So this is mainly when you're in coastal regions. What we did find, we don't really know what it means yet. So these long strings are fecal pellets from krill. And all these small organisms are Microcetella sitting on them. So we don't know if they use fecal pellets for food so much, or if they use them as a highway to go back down and then they continue their search up. But that's a study for later or for somebody else. So basically what we concluded was that these 33% could easily be explained by zooplankton feeding during night and in different regions during the polar day. Then we had the 13%, which happened both during day and night. So during day, we found 30% attenuation. We assume that this is microbial degradation, which shouldn't really change between day and night. And it also happens in the water layer below. It's more or less the same. So my PhD supervisor, Helle Plough, taught me how to use her flow chamber, where we can use a microsensor, which is what you see here, to measure the gradient of oxygen in the few hundred micrometers at the boundary layer around a small marine snow aggregate. And we can then use this gradient to calculate the total oxygen consumption, which we then convert into CO2 production and can use it to calculate the microbial degradation of these aggregates. So we do it in a flow chamber where we suspend the aggregate. So we add a flow that matches the settling velocity of this aggregate. And then we use the microsensor to measure a gradient through the aggregate of oxygen. And we then use Fick's law and this boundary layer to really calculate what the respiration is. So basically the oxygen consumption or the flux of oxygen into the aggregate, convert that to the flux of CO2. Again, here we need a conversion factor, which is somewhere between 0.8 and 1.2 from oxygen respiration to CO2 reduction. And when we do that for all kinds of aggregates in all kinds of regions, we found that on average, it's around 13%, which fits quite nicely to what we also measured in situ. This temperature, so this is at 15 degrees. This changes a lot with temperature, of course. We haven't done it with pressure, so pressure might have a bigger impact than we think, but that's something we really want to also do in the future. So basically, we assume that a lot of it is microbial degradation, happens day and night, and then we have this additional nightly zooplankton grazing for the attenuation in the upper water column. And then the question is, why is it so much lower in the deep ocean? Because you would expect if this is colonized by deep ocean bacteria, they are accustomed to high pressure and to whatever is down there. So first we did a little test to see is it just temperature or is temperature really important? So we took aggregates at 15 degrees, formed them, measured carbon-specific degradation, took the same aggregates with the same microbes, added them to 4 degrees, measured the carbon-specific degradation again, and then we compared it to the in situ. And we found that they were pretty much the same. There was 3.5 fold reduced compared to at 15 degrees, which gave basically a Q10 of around 3.5. So when you reduced temperature by 10 degrees, you had 3.5 fold reduced carbon-specific degradation. And in the past, it was assumed, even though in Kerber's paper, he also said that some bacteria were trapped in the matrix of an aggregate. But basically, we believed that aggregates would only, bacteria would only stay on an aggregate for 20 minutes and then they would jump off. I think nobody believes that anymore, but that was where we started. And assuming that deep sea bacteria are efficient at low temperatures, we can't really explain this if we have this jump on and off. And there have been a few studies testing this, and I think the best one I have been involved in is by Eddie Fadeve, who was my former PhD student. He's now working with Gerhard as a postdoc. And here we were comparing, or we were classifying the microbial community in different depth layers. So you can see the colors down here. The light blue is the surface ocean. The somewhat darker blue is the epipelagic, and then we have the mesopelagic and the deep ocean. So we did that for the free living bacteria, and then we collected settling particles and did the same for those. And in the surface, we saw that most of the microbial community was basically what was in the surrounding water. We could explain it. But the deeper we got in the water column, we saw that we still had a large fraction of the surface bacteria there, even though it did decrease. But we didn't really get a large fraction of the community from the epipelagic into the aggregate, or the mesopelagic, or the basopelagic. Instead, we got a large unknown fraction, which we think is still the rare fraction that was in the surface, but as an aggregate sink changes its organic matter composition, pressure, temperature, etc., then this rare fraction has a chance to actually bloom and become more important. But it's not that we have a strong jumping on and off of these. So basically all the bacteria originate from the surface ocean, and as they sink down, encounters higher pressure, colder temperature, different organic matter composition, the activity becomes less efficient. And that is probably why we have this decrease in microbial degradation. So temperature, pressure, but also maybe organic matter composition. So we basically found that attached microbes within settling aggregates in the deep ocean seem inherited from the surface ocean, and there's only little colonization and detachment at depths as these aggregates sink down. We wanted to test that, so we took a global ocean data set from sediment traps. We only included sediment traps that were below 1,000 meters. So the upper trap should be below 1,000 meters, where it's 4 degrees less. And we needed a deeper sediment trap to include it, which was collected around the same time and is in the same area. And then we were trying to calculate this, basically carbon-specific degradation, you could call it, by taking the upper trap and doing the L ratio, which is basically the relationship between degradation and settling velocity. So it's the carbon-specific degradation per meter settled over the distance settled. So this is basically the fraction of organic matter lost multiplied with what the flux was for the upper trap, and that should then give us the flux at the lower trap. When we did that, we found that if we use the degradation rate at 15 degrees, then this is the regression we got. And the solid line is what we actually measured, and the 4 degrees is this dashed line. So we found, ignoring pressure effects and ignoring organic matter changes, we could explain basically the flux to the deep ocean by only taking temperature decrease and decrease in carbon-specific degradation rate as a function of temperature into account. I'm not saying that this is the golden way, there's definitely other processes, but it was very surprising how well they fitted and they had the same slope. So temperature definitely seemed to be a major component in the decreasing flux attenuation in the deep ocean. This is temperature limitation could explain deep ocean flux attenuation, and that was due to trapping of surface microbes into settling aggregates, and without an increase, and this was done without increase in pressure or organic matter transformation. So I wanted to check, why is it that microbes doesn't attach to sinking particles? So we collected in situ marine snow, put them in to the flow chamber again, but added a laser light and suspended particles to look at particle imaging velocimetry. And in this way, we could really measure the boundary layer around an aggregate. So you have a settling aggregate which has this boundary layer of very, very slow flowing water. And that means you have basically this border at an aggregate. So in order to cross it, you need to be a swimming aggregate that can actually cross it. And the deep ocean, there are swimming aggregates, but it's, I think it's 20% or something. Gerhard can probably correct me afterwards. It really means that it's only a smaller fraction of the deep ocean bacteria that even has the potential to get close enough to an aggregate to attach to it. So just to do a fast summary, I can see I'm already running out of time. So we now know that we have fixation of CO2 into organic matter by phytoplankton in the surface ocean. We have different kinds of phytoplankton, phylloxystes, diatoms, coccolithophores. Some have ballast minerals, some doesn't. So that means we are forming fast and slow sinking aggregates. Then we have the zooplankton that feed on the aggregate, that disaggregate the aggregates, and in total they add up to the slow sinking. They also produce fecal pellets, which can sink fast, which can then be fed on by microbes, either directly by flagellates or the whole microbial community. And it's mainly the fast settling aggregates that actually make it out of the surface ocean. Here we have limited degradation, possibly due to temperature. And if we put some numbers on, we have 30 to 70% of the attenuation is due to zooplankton, 3 to 13 due to microbes, and in the deep ocean is around 3% microbial carbon-specific degradation. So that means if you look at it vertically, we have primary production in the surface aggregates, so each settling aggregates. Okay, now I have to see if I remember. No, this waits a little bit. So we have the primary production. Below that depth, we have aggregate formation. Then we have filter feeding, which produce fecal pellets and vertical migration. We have a lot of microbial organism that feeds on the small aggregates that are sinking slowly. We have aggregate feeding, aggregate fragmentation, fecal pellet production, coprophagy, which is basically feeding on settling fecal pellets, and then we have microbial degradation. This is how I kind of see it in the upper water column of carbon flux attenuation. But then we also have to consider an aggregate is not just phytoplankton and bacteria. It's a whole microcosm of. So when we are measuring respiration of an aggregate, it's not just bacterial respiration, it's also phytoplankton respiration, ciliates are respiring, nanoflagellates, dinoflagellates, etc. So everything is respiring inside this aggregate. And the bacteria are producing nutrients, dissolved organic carbon, the nanoflagellates produce CO2, they produce dissolved organic carbon as well. We have ciliates, and so we have all these organisms. And at the same time, we are changing detritus into animals, so into proteins basically. And here, this is from an in situ collected aggregate where you can see ciliates and nanoflagellates. You will see here, and so you can see it's not just bacteria and phytoplankton. It's a whole ecosystem inside these aggregates. So when they're sinking down, you also get the zooplankton feeding on them. And what we really find is typically in fecal pellets and aggregate fragments, you have high carbon to nitrogen. So it means when you get all these trophic levels, you basically get that you're sinking out carbon-rich but nutrient-poor aggregates, and you leave the nutrients behind in the surface. So remineralization reduces the magnitude of carbon export, that's true. Technically, it reduces the efficiency of the carbon pump, but heterotrophic activity recycles nutrients over carbon and promotes carbon-rich aggregates to fertilize the surface ocean and extend the plankton bloom. So basically, you have all these recycling going on, and that really extends the whole recycling and the primary production for a much longer time and makes for a much more efficient carbon pump because primarily carbon sinks out. So though biological activity reduces immediate carbon flux, it retains nutrients in the surface ocean and promotes export of carbon-rich aggregates. And if you view it for the whole productive season, I would argue that biological activity increases the efficiency of the carbon. And I think we just skip over. I'm getting really late now, but we saw that we actually had a lot of iron in the Southern Ocean reduced from krill pellets, or in the tropical regions, we have a lot of nitrogen recycling happening in these aggregates. And when we modeled it, we also saw when we included macrozooplankton, we got a much more efficient carbon pump around the Southern Ocean. So it seems that nutrient recycling via zooplankton creation is important for both primary production and carbon export. And I think we're basically there. So the idea is we need to really not just measure POC, to understand how microbes and zooplankton transform organic matter as it sinks, and what is the role of zooplankton versus microbes for this nutrient recycling. That's it.